Hello everyone. Today we are going to see the topic PCR. PCR is nothing but the polymerase chain reaction. So this presentation introduced the students to clear more about the knowledge on the polymerase chain reaction, which is very important technique in molecular biology, where it can amplify the specific DNA fragments mainly in tool in diagnostic research and biotechnology. So it is mainly designed for the microbiology students who are very eager to get a proper understanding of the PCR and its principles, procedures and its applications. So coming to the introduction, PCR, it is a laboratory technique which is used to produce the million copies of specific DNA. So if one DNA it gets amplified into million copies only from the small sample of DNA. So it was introduced by the scientist Mullis in 1983, who was rewarded for the Nobel Prize in Chemistry in 1993 due to its transformating impact on the science and molecular biology. And this technique is widely used in medical diagnosis and in the field of genetic research, and it also helps the scientists to work with minute quantity of DNA for research purposes also. And coming to the principle, it is based on the natural process of DNA replication. So DNA replication, in the sense the DNA gets multiplied into 2, 4, 8, 16, 32. So it is the controlled amplification in the laboratory setting, and this technique mainly uses the thermostable DNA polymerase, typically called as Taq polymerase. So this DNA amplification is done mainly with the enzyme Taq polymerase, which is derived from the thermophilic bacterium. So thermophilic, in the sense, thermo means heat, philic means loving. So it is heat loving species, that is Thermus aquaticus. So this species of bacteria can withstand the high temperature without denaturation. So without denaturation, in the sense it does not denature into single pieces, so it can withstand that amount of high temperature. And this PCR involves the thermal cycling. So thermal cycling, in the sense, the process of repeated heating and cooling process. So one process it gets heated and the other process it gets cooled. So continuous heating and cooling process has taken place in this PCR polymerase chain reaction. So this PCR is mainly done with the help of thermal cycler. This is the thermal cycler machine in which a PCR has to be run in this machine. So you could see the parts of sample chamber closing lid and sample vials chamber. So in that sample vials chamber only, we want to fix that our PCR tube, which is filled with some of the buffer solution, we could see later, and digital display is there, and time and temperature adjustable keyboard is also there, and time adapting buffers also there, and the heat releasing parts are also there. For if suppose more amount of heat is generated, it gets released off from that machine for the prevention of the thermal cycler machine. So this is the overall condition of the thermal cycler, in which the PCR and the amplification of DNA takes place in the thermal cycler machine. So coming to the buffers and reagents used. So for normal molecular biological techniques, we have some of the buffers and reagents in that reaction for enhancing its specificity and enhancing its sensitivity of the results. So coming to the PCR, the following buffers or reagents shall be added. First one is main important is template DNA. So template DNA is nothing but it's the original form of DNA in which that DNA has to be amplified in that PCR. So template DNA, original DNA is must. And primers. So primers is nothing but the short strands of DNA sequences, which typically consist of 18 to 22 nucleotides. So nucleotides, in the sense, are DNA is composed of nucleotides, right? Adenosine, guanine, time in cytosines. Likewise, nucleotides are building blocks of DNA molecules, which is composed of deoxyadenosine, deoxythymidine, deoxycytidine and deoxyguanosine triphosphate are used to mainly construct the new DNA strands from that original DNA strand. And as I said before, DNA polymerase enzyme, this enzyme paves for the synthesis of new DNA strands by adding nucleotides to the primer. So these are the main buffers and reactions which is used for running the PCR cycle. And moreover, you could see the PCR tube, and this PCR tube is in this PCR tube only we are going to adding the buffers and reagents and placed in the thermal cycle for the reactions to be carried out.